Hey everyone, and welcome back to BiochemSerye Learning on the Go. We're on episode 5 now, and you know what that means. It's time for us to dive into the chemistry of carbohydrates.
Mm,
You can think of us as your personal biochemistry tutors, here to simplify all those concepts that might have seemed a bit overwhelming back in class.
that's right. We're here to demystify these sugars, make them a little less daunting, maybe
A
even...
little more digestible.
Exactly.
You're in for a real treat today because our expert is not only a fellow professor, but a true carb enthusiast. Well, at least when it comes to biochemistry, that is.
Well, somebody's got to explain to those med students why they can't survive on donuts alone, no matter how tempting they are.
So true, so true. Okay, let's start with the basics. What exactly are carbohydrates and why should we, or any student for that matter, care about them?
Well, carbohydrates are absolutely essential molecules. You could even say they're the building blocks of life. They're made up of carbon, hydrogen, and oxygen.
Hence the name.
Exactly, hence the name. And their primary role is to provide energy for our cells, but it's not just about energy, though.
Oh, that's right. I remember teaching about how they're also involved in structure and communication between cells, and that always blew my students' minds.
It does, doesn't it? It's like they're the ultimate multitaskers of the biomolecule world. You can think of a house, right? You've got the framework, that's like structural carbohydrates. Then you've got the electricity running through it. That's the energy from carbs. And then you have the people living in the house communicating, which represents cell signaling.
I love that analogy. That's great. Now, let's break it down even further.
Okay.
We always start with the building blocks.
Hmm.
Monosaccharides, disaccharides, and polysaccharides. What are they and how are they related?
You're right on track. You're right on track. Monosaccharides are your simplest sugars. Think mono, meaning one. They're the ones that you can't break down any further. They're like those individual Lego bricks that you use to build all sorts of amazing carbohydrate creations.
So they're the foundation, the single units.
Yeah.
I always tell my students to think of glucose as an example. That's a big one they'll encounter repeatedly.
Absolutely. Glucose is the star of the show, but you've also got fructose and galactose, each with its unique properties. Now, each of these bricks has specific features like a carbonyl group, which can be an aldehyde or a ketone, and multiple hydroxyl groups. And it's these subtle chemical details that really give each monosaccharide its own distinct personality.
Now, I remember this always tripped my students up.
Oh, yeah.
Chirality, can you explain that in a way that even a first year wouldn't completely freak out about?
Sure. Chirality just means that a molecule can exist in two forms that are mirror images of each other, like your left and right hand. They have the same parts, but they're arranged differently in space. And here's the thing. This tiny difference can have a huge impact on how a molecule behaves in biological systems.
It's like trying to put a left shoe on your right foot. It's just not going to work.
Exactly. Exactly.
So how does this play out with monosaccharides?
Well, monosaccharides have these chiral carbon atoms, meaning they can have different isomers, those mirror image twins.
Right.
And even though they might look similar on paper, these isomers can have completely different effects in the body. Like, take glucose, for instance. It's essential for energy, but its isomer, galactose, is part of lactose, which is the sugar in milk.
So chirality is kind of like a subtle molecular switch that can totally change a sugar's role. That's a good way to explain it to students. Now, what happens when we combine these single sugar units, that's where we get into disaccharides, right?
Exactly. Two monosaccharides joined together, that's your disaccharide. Think of it like snapping two Lego bricks together. The connection is a special type of bond called a glycosidic bond.
Ah, yes, the infamous glycosidic bond. How do you explain that one without making everyone's eyes glaze over?
Oh, that's easy. Imagine making a sandwich. You've got your two slices of bread. Those are your monosaccharides. And then you have your filling. That's your glycosidic bond holding everything together.
I like it. So what are some of the superstar disaccharides we should all know about?
Well, you've got sucrose, the common table sugar we all know and love. That's glucose and fructose teaming up. Then there's lactose, the sugar found in milk, which is glucose and galactose. And finally, maltose, which is formed from the breakdown of starch. It's two glucose molecules bonded together.
So disaccharides are like those double-decker Lego creations. What happens when we go even bigger into the world of polysaccharides?
Polysaccharides are the real heavy hitters. These are long chains of monosaccharides, all linked by, you guessed it, glycosidic bonds. They can be hundreds or even thousands of sugar units long.
Wow, I always picture it like those super long paperclip chains we used to make as kids. except instead of paper clips, it's all sugar molecules.
That's a great way to visualize it. These giant molecules play crucial roles in energy storage and building structures. Like, for instance, plants store their energy as starch, while animals use glycogen for the same purpose.
Right. So plants have their pantry stocked with starch, found in things like potatoes and rice, and we have our glycogen reserves, mainly in our liver and muscles. It's just fascinating how different organisms have evolved these distinct ways of storing their sugary fuel.
It is, and when it comes to structure, we have to talk about cellulose. It's the most abundant organic compound on Earth, and it forms those tough cell walls in plants. We can't digest it, but it's super important for us as dietary fiber, keeping our digestive system running smoothly.
So cellulose is like the plant's armor, giving it strength and rigidity.
Yeah.
And even though we can't digest it, it's still essential for us as dietary fiber,
Yeah, it is.
keeping our digestive system in tip-top shape.
Exactly. Now, speaking of tough structures, let's not forget about chitin. That's the main component of insect exoskeletons and the cell walls of fungi. Talk about a strong material.
So it's clear that from powering our cells to building the very structures of life, carbohydrates are pretty incredible.
They are. They really are. And we've really only just scratched the surface here. There's a whole world of specialized carbohydrates out there, like glycosaminoglycans or JAGs. Yeah, these are super important in connective tissues. And then we've got glycoproteins, which are proteins that have these carbohydrate chains attached. They play vital roles in cell recognition and signaling.
It's amazing how these little sugar molecules can be involved in so many different processes. It's like they have a hand in everything our bodies do.
They really do. And the more we learn about them, the more we realize just how crucial they are. But before we jump into those specialized carbohydrates, I think it's important to take a moment to really appreciate the incredible diversity of these molecules. From single sugar units to these massive polymers, they're involved in energy, structure, communication, and so much more. It's truly a testament to the elegance and complexity of biochemistry.
Well said, and I think our students and our listeners would agree. Carbohydrates are a lot more exciting than they might have initially thought. And I have a feeling this is just the beginning.
It is. You know what always gets me?
What's that?
Is how these sugar molecules aren't just static. You know, they're not just sitting there. They're dynamic. They react and change. Depending on their environment, it's like they have a whole repertoire of moves.
It's like they're dancing. I remember when I was teaching reactions like mutarotation and glycoside formation, my students would look at me like I was speaking another language. Maybe a dance analogy would help.
Maybe. Okay, so let's think about it this way. Mutarotation is all about those anomers we mentioned before, the alpha and beta forms. Remember how those hydroxyl groups can kind of switch positions at the anomeric carbon?
Yeah.
Well, in solution, those sugars are constantly opening and closing their ring structures, flipping between those alpha and beta forms, like they're doing a little twist or a turn. And as they do that, they change their optical rotation.
So it's not just a fixed structure. It's like this dynamic equilibrium.
Yeah.
Which I know can be a really tough concept.
It is.
For students to really grasp.
Yeah. Now, with glycoside formation, that's where we're building those glycosidic bonds, those links in our sugar chains. It's almost like those sugar molecules are reaching out and grabbing onto another molecule to form a new connection, and that other molecule could be another sugar, or it could be an alcohol, maybe an imine.
I always tell my students to think of it like they're building a Lego tower. Each brick is a monosaccharide. And then that glycoside formation is the act of snapping those bricks together.
Perfect analogy. And you know those glycosidic bonds are essential for building everything, from your simple disaccharides to those complex polysaccharides and even glycoproteins.
Now, speaking of glycoproteins,
yes,
those always seem to be a big aha moment.
Yeah.
For students when they realize how sugars and proteins can actually team up.
They do.
To carry out these amazing functions.
Absolutely. But before we get too deep into those, let's touch on some other reactions that our sugars can undergo. Oxidation and reduction are key players in the carbohydrate world.
Yes, those two always caused a bit of confusion, even in my upper level classes.
Oh, yeah.
It's like those electrons are constantly playing this game of hot potato.
You got it. Oxidation is losing electrons, reduction is gaining them. And our sugars can be oxidized or reduced at different spots, leading to all sorts of interesting derivatives. For example, the oxidation of glucose can produce gluconic acid, which is actually a common food additive.
I never knew that. See, there's always something new to learn about carbohydrates.
Always. And then on the other hand, the reduction of glucose can produce sorbitol, which is a sugar alcohol often used as a sweetener.
So these reactions are like giving those sugar molecules little makeovers,
Yeah.
changing their properties, giving them new roles to play.
Exactly, exactly.
And what about epimerization? That one always seemed kind of abstract to me.
Right, so epimerization is all about those epimers we talked about earlier, those isomers that differ at only one chiral center. It's like flipping a molecular switch, converting one epimer to another.
So are you saying glucose and galactose, which only differ at one chiral center, can actually be converted to one another through epimerization?
That's exactly right. And we have these enzymes called epimerases. They're the masterminds behind this switch, and they play crucial roles in carbohydrate metabolism, making sure that we have the right sugars at the right time.
It's amazing how our bodies have these perfectly tuned mechanisms to control all these reactions.
It is. It is. And let's not forget about esterification, another important reaction for our sugars.
Yeah.
So here, an esco bond is formed between an alcohol group, often from the sugar, and a carboxylic acid group, almost like adding a fancy pendant to our sugar molecule.
I love that. So do these sugar esters then have special functions in the body?
Oh, they do. For example, glucose 6-phosphate, a sugar ester, is really important in glucose metabolism. These esters are like actors in a play. Each one has a specific role in the grand scheme of biochemistry.
Now, speaking of roles,
Yeah.
let's shift our focus to those amazing plant polysaccharides we were talking about before.
Yeah.
We covered starch, which is how plants store glucose. But what about those structural powerhouses, cellulose and hemicellulose?
Ah, yes. Those two are absolutely essential for giving plants their shape and their strength. Cellulose is the most abundant organic compound on Earth.
Wow.
It forms those rigid cell walls that we as humans can't digest, but we rely on for fiber.
Right. I always picture cellulose as kind of like the scaffolding that holds the plant upright.
That's a great image. Now, hemicellulose, which is a diverse group of polysaccharides, it's like the glue that binds those cellulose fibers together to provide that extra support.
So it's like the mortar between bricks, adding strength and flexibility to the plant's structure. Now, structurally, how do these two differ?
That's a good question. So cellulose is a long linear chain of glucose units linked by beta-1-4 glycosidic bonds. These bonds create these straight, rigid chains that pack together really tightly. To form strong fibers, now hemicellulose on the other hand, is a bit more of a mixed bag.
Okay.
It's made up of various sugar units and has both linear and branched regions.
So it's like cellulose is a perfectly ordered stack of logs, while hemicellulose is more like a jumbled pile of branches, adding complexity to that overall structure. Now, I know from teaching nutrition that humans can partially digest hemicellulose, but not cellulose.
Yeah.
Why is that?
It all comes down to those glycosidic bonds, you see. The enzymes in our digestive system, they can break down the bonds in hemicellulose, but not those beta-1-4 glycosidic bonds in cellulose. So that's why cellulose is considered dietary fiber. It just passes through our digestive system pretty much intact.
It's like cellulose is the guest that shows up to the party but doesn't eat anything.
Yeah.
And hemicellulose is the one who mingles and enjoys all the snacks.
I like that analogy. Now, while we're on the topic of plant polysaccharides, let's not forget about pectin.
Oh yes, pectin.
It's a gel-forming polysaccharide found in the cell walls of fruit.
Pectin, the reason we can enjoy our jams and jellies.
Exactly.
Like the molecular magician, it can turn liquids into solids.
It is, and pectin actually has health benefits too. As a soluble fiber, it can help lower cholesterol and regulate blood sugar.
So it's not just delicious, it's good for you too. Now let's talk about those fascinating glycosaminoglycans, or GAGs that we mentioned before. They're the ones that help keep our joints moving smoothly.
You got it. GAGs are a really diverse group of polysaccharides with a unique structure
Okay.
and a really wide range of functions.
Let's break it down. What makes them so special?
So GAGs are these long, unbranched chains composed of repeating disaccharide units, and typically these units include an amino sugar, like glucosamine and a uronic acid. Now, what's really interesting is that a lot of GAGs are sulfated, meaning they have sulfate groups attached, which gives them an increased negative charge.
So it's like they're all decked out in these negative charges, ready to party.
Exactly. And, those charges are actually really important for their functions.
How so?
Well, GAGs are found in a variety of connective tissues, everything from cartilage to bone, skin, and blood vessels. They provide structural support, lubrication, and hydration.
It's like they're the ultimate multitaskers of the connective tissue world.
Yeah.
Now, I know there are different types of GAGs. What are some of the big names we should know?
Oh, there's so many. Well, there's hyaluronic acid, the one everyone's buzzing about these days. known for its ability to hold massive amounts of water,
Oh yeah, hyaluronic acid.
making it a superstar in skincare and joint health. And then we've got chondroitin sulfate. Another important component of cartilage, provides structural support. And it's often used as a dietary supplement to help ease joint pain.
So hyaluronic acid is like the hydrating hero,
Yes,
and chondroitin sulfate is the cartilage champion.
that's a good way to put it.
Are there any others we should be familiar with?
Oh, absolutely. We can't forget about heparin, a gag known for its anticoagulant properties, meaning it helps prevent blood clots. It's used medically to keep those blood vessels flowing smoothly.
Wow, so gags are involved in everything, from keeping our skin plump and hydrated to making sure our blood flows properly. They truly are the unsung heroes of our connective tissues.
They are. They really are. Now, before we completely wrap up this section on carbohydrates,
Okay.
we have to touch on those glycoproteins and glycolipids that we mentioned earlier.
Oh, yes.
Remember, they're all about cell recognition and signaling.
They're the ones with those sugar chains, acting like molecular name tags and gatekeepers for our cells.
Exactly. Essential for a wide range of biological processes. Everything from immune responses to fertilization.
So let's dive into the details.
Okay.
What makes glycoproteins and glycolipids so unique?
Well, they're formed by attaching carbohydrate chains called glycans to proteins and lipids, respectively. These glycans are like molecular codes, providing cells with crucial information about their identity and function.
It's almost like they're wearing little barcodes,
Mhm.
allowing cells to scan and recognize each other.
That's a great analogy. Now, glycoproteins are found everywhere, in cell membranes, the extracellular matrix, even in blood. They're involved in cell adhesion, signaling, immune responses, and even fertilization.
It's like they're the social butterflies of the cellular world interacting with everyone.
They are. Now, glycolipids. They're mainly found in cell membranes, where they play crucial roles in cell recognition, signaling, and membrane stability.
They're like the gatekeepers of our cells, deciding who gets in and who stays out.
Exactly. And they're also really important for the nervous system. They form that myelin sheath that insulates nerve fibers.
Wow, so they're involved in communication and protection.
Absolutely. And here's what's really fascinating.
What's that?
Those glycans on glycoproteins and glycolipids, they can vary a lot in terms of their structure.
Okay, let's talk about glycan diversity then. What factors contribute to this variability?
Well, you've got the types of sugars that are present, how they're linked together, and modifications like sulfation and phosphorylation. It's like a molecular mix and match, creating this huge array of glycan structures.
So that they're speaking different languages, conveying specific messages to cells.
Exactly. Now, let's look at some specific examples of glycoproteins and glycolipids.
Okay, I'm ready to meet these molecular celebrities.
All right, so one famous glycoprotein is the ABO blood group antigens. These are found on the surface of our red blood cells and determine our blood type.
Oh yeah, the ones that tell us who we can donate blood to and
Exactly.
receive blood from.
They differ in their glycan structures, and these differences can actually trigger immune responses if incompatible blood types are mixed.
It's like a molecular handshake. If the handshakes don't match, the immune system sounds the alarm.
Exactly. Another important glycoprotein is immunoglobulin G, or IgG for short,
IgG.
the most abundant antibody in our blood.
IgG, the antibody superhero fighting off infections.
That's it. And IgG has these complex glycans that are super important for its function. They enhance its ability to recognize and neutralize those nasty pathogens.
It's like those glycans are the antibody's secret weapon. Now, for glycolipids, let's talk about gangliosides.
Yes, gangliosides.
These are the brain's sugary companions. What do they do?
They're found in high concentrations in the nervous system, and they play essential roles in nerve cell development, signaling, and function. It's like they're the brain's communication network, making sure all those signals are transmitted properly.
That's amazing. So these examples really show us just how diverse and essential glycoproteins and glycolipids are. From determining our blood type to ensuring our brain functions correctly, they play such a vital role in maintaining our health.
They do. Now, before we move on, let's just take a moment to acknowledge this booming field of glycobiology,
Oh yes, glycobiology.
the study of all things sugar.
The science of sweetness. It seems like we're only just beginning to understand the complexity.
We are, and with each new discovery, we're uncovering potential new targets for drug development and disease treatment.
It's like unlocking this treasure trove of therapeutic possibilities.
Exactly. The world of carbohydrates is full of wonder, and as we continue to explore it, I'm sure we'll find out even more about these fascinating molecules.
Well said. And before we wrap up this part of our carbohydrate journey, I just wanted to add a personal note. As professors, we often see our students struggling.
You do.
With the intricacies of this subject. But I hope that this deep dive has shown that carbohydrates are more than just memorizing structures and formulas. It's about really understanding the intricate and fascinating ways that these molecules contribute to life itself.
I completely agree. From energy production to cell signaling and immune responses, carbohydrates are the unsung heroes of our cells, working tirelessly behind the scenes to keep us functioning.
And their study offers such a unique glimpse into the elegance and complexity of biological systems.
It does. It does. Now, are you ready for the quiz portion of our show?
Yes.
I know you've been waiting to test our listeners' knowledge.
I have. Let's see what they've learned about the sweet, sweet world of carbohydrates.
All right, bring it on.
Okay, so you've traveled with us through this amazing world of carbohydrates, from those basic sugars to the complex polymers that are really the building blocks of life. Now it's time to see what you've learned. Are you ready for a little carbohydrate quiz?
Hit me with your best shot. Let's see if our listeners have been paying attention.
All right, first question. What is the primary function of carbohydrates in living organisms? This one's easy.
It is. It is. It's to provide energy for cells, Carbohydrates get broken down to produce ATP, which is the molecule that powers almost every process in our bodies.
Exactly. Next question. What's the main difference between aldoses and ketoses?
Think about where that carbonyl group is located. In aldoses, it's at the end of the carbon chain, forming an aldehyde. While in ketoses, it's within the chain, forming a ketone.
You got it. Okay, onto a specific sugar. Which monosaccharide is commonly known as blood sugar?
That would have to be the famous glucose, the VIP sugar that travels throughout our bloodstream, delivering energy to all our cells.
That's right. Moving on to bonds. What kind of bond links those monosaccharides together to create a disaccharide?
The glycosidic bond. And it's formed through a dehydration reaction. Picture two hands clasping together, and a water molecule is squeezed out in the process.
Perfect analogy.
Yeah.
All right, let's test your memory. What is the main storage polysaccharide found in plants?
That would be starch. It's at the plant's pantry, all stocked up with glucose, ready to provide energy whenever it's needed.
And just to add to that, what's the difference between amylose and amylopectin? Those two components of starch.
Good question. Think of amylose as a straight chain of glucose units, while amylopectin is more like a branched tree. And this difference in structure actually affects how quickly they can release that stored energy.
Excellent point. Now let's switch gears to animal storage. What's the storage polysaccharide found in animals?
That's glycogen, mostly stored in the liver and muscles, acting as your backup energy supply, ready to go when you need a quick boost.
Correct. Let's talk about structure now. What's that incredibly abundant organic compound found in plant cell walls? The one that makes plants so strong?
We're talking about cellulose, a tough, fibrous polymer of glucose. We humans can't digest it, but it plays a vital role as dietary fiber, keeping our digestive system happy.
Exactly. Now for something a little tougher, what are those long, unbranched polysaccharides that we call GAGs, and what's their main function in our bodies?
Okay, so GAGs are like the cushions and lubricants in your connective tissues, providing support, padding, and keeping everything hydrated. They're negatively charged, often with sulfate groups attached, and they attract water like crazy.
You got it. Last question. What are those interesting molecules called glycoproteins? What roles do they play?
All right, so glycoproteins are basically proteins, all dressed up with carbohydrate chains. Think of those chains as molecular name tags. They're essential for cell recognition, signaling, and those all-important immune responses.
Fantastic. You aced the quiz. Give yourself a big round of applause. It's been such a fantastic deep dive into the world of carbohydrates.
It has. From simple sugars to those complex polymers, these molecules are crucial for life. We've gone over their structures, functions, reactions, and even that exciting field of glycobiology.
Absolutely. And I hope you, our listeners, have gained a deeper understanding of the amazing world of these sweet and complex biomolecules.
Definitely remember, the world of biochemistry is just full of wonders waiting to be discovered.
Now that you've got a good grasp on the fundamentals of carbohydrates, we want to leave you with something to think about. We often hear about good carbs and bad carbs, but is it really that straightforward? Are all carbs created equal? That's a question we want you to explore as you continue your journey through the world of biochemistry. Keep asking those questions and stay curious.
Until next time on BiochemSerye Learning on the Go, happy learning, everyone.